Yeah, good morning. It's my extreme pleasure to have NCI Designated Cancer Center Director as our speaker today. We don't have that terribly often. A couple of things about Howard. He said, keep it short. So he grew up in North Dakota near Fargo-Moorhead. And like a lot of people that want to move south to a climate that's a lot warmer, he finished medical school and moved to Madison. So that's about the same. It's like us going to Fort Myers or something like that, I guess, about the same differential in temperature. But in any case, he's been at Madison for his whole career. He was very active in the NCI RAID program when that was active back in the late 80s, early 90s. And he worked with Paul Carbone, for whom the center's named. And he worked his way into cancer prevention and had a series of studies that were very important to be accomplished. And I got to know Howard about 10 or 11 years ago now. He's one of the 5 or 6 NCI consortium holders for this large collective cooperative group, if you will, of how the bread and butter of prevention clinical trials get done for the last decade or so. And prevention is always that holy grail. But what I've personally found is that anytime I spend 5 minutes or so with Howard, or a few more minutes than that, there's always going to be some kind of a conventional wisdom that comes out about certain things, about accrual to a prevention trial, different ways to try and optimize things. And we still don't do the best of jobs with this. We don't have people beating down our doors for prevention trials, but that's really where the rubber meets the road for all the preclinical work that we do in carcinogenesis. And it's the absolute necessary part of the pipeline. And it was last year we had Dr. Steele here, and to follow him up, it's wonderful to have Dr. Bailey here as well. So I'm going to let him start.
Thank you, Frank. Again, it's my pleasure to be here at the University of Minnesota and talk to you a little bit about cancer prevention. And as the title talks, I'm going to use kind of the nuts and bolts of this Cancer Chemoprevention Consortium and some of the studies to give an examples of the struggles we have in cancer prevention agent development with it. So in terms of disclosures, recently there's been a fair bit of press in the cancer literature about people not disclosing all the things. They just happen to forget that they were making $1 million from this company or that company. So I thought I'd go overboard on that. And so the funding from the NCI, and the other reason I thought is since I can be influenced by a free can of Mountain Dew, I thought I should probably give you all the things that are potentially here. But so biomarker science, and actually many of these, or a couple of these I haven't even received funds for, but someday I might on their advisory board. We'll see what happens with it. But so what I hope to talk about today and describe to you is really using the studies that we're doing as, again, given examples of the issues, struggles in cancer chemoprevention or cancer prevention agent development. And here's just the agenda. And also, despite the more than decade that you've been involved in the consortium, to give you a little bit more background on it, something that Frank and many others have been very critical to our success related to that. So why talk about cancer prevention? Well, as that wise sage, Ben Franklin, said, an ounce of prevention is worth a pound of cure. So there's certainly that rationale for it. But there's also no getting around that the study of carcinogenesis, much of that work done here, and as well at Wisconsin and other places, really comments on and shows that it's a many-year process. And of course, that's not really unique to any number of health maladies we deal with, whether it's vascular disease or pulmonary disease, whatever. The fact that the disease development is measured, or the genesis of it is over years, gives us opportunities to intervene. And if we show it on a kind of a figurative thing here, and this is, again, just a graphical model. It could be breast tissue, pulmonary tissue, whatever. The fact that our thoughts are that this dysplastic, this nuclear atypia, the tissue changes that occur, the genotypic followed by phenotypic changes, take years normally. Now, again, there's some data from some researchers that raise questions that there might be unique individuals where this process is sped up and is much shortened, but the vast majority really is measured in multiple years, thus giving us, and again, the key thing being from a clinical standpoint and from a study standpoint is that cancer and invasive disease is that. It's the ability to invade. That, again, nuclear atypia, genotypic changes, all that stuff it all comes down to the difference between malignancy and non-malignancy is this invasiveness, and that's ultimately what we're trying to prevent. So with that in mind, what I thought I'd start with is just giving you a list, and there's 3 slides here that show the current, what I would call FDA-approved agents, interventions related to diminishing cancer incidence. And when you look at the far right, FDA indication, some of the key things with it is looking at that. So tamoxifen and raloxifene for breast cancer are fairly straightforward. Celecoxib and FAP disease. But BCG and valrubicin are really getting into the treatment of superficial bladder cancer. Now here again we're getting into more reduced incidence, in this case of infections, oncogenic infections with the HPV vaccines. And I make note of, and I have read in the recent change in the population that's approved for Gardasil 9, or the 9-valent vaccine, which is the age increase now up to age 45 for men and women. And then this last slide, which really depicts kind of some of the issues, which it's really getting into the treatment of Barrett's esophagus or actinic keratosis. And when you look at all of these agents here, and this kind of summarizes it. When you really get right down to it, the vast majority of things that are out there that are on this list really came about in the ways as summarized. Either treatment of existing conditions, actinic keratosis, the scaly redness, the cosmetic issues that are involved with it, or Barrett's esophagus. Now again, Barrett's, we have always known and worried of the potential of it becoming invasive disease. But tamoxifen and raloxifene really were discovered through therapy studies. That through our adjuvant studies, we found that contralateral breast cancer incidence was much reduced, which then led to the prevention studies. That, and then I'm not going to take the time to summarize it, but many of you already know it, that when we did some of the larger phase 3 prevention studies, whether it's selenium and vitamin E in prostate cancer, whether it's STAR or the tamoxifen studies, they really went straight from either epidemiologic data or other data right into these. And SELECT, while we learned a lot about prostate cancer, the unfortunate truth is we spent hundreds of millions of dollars to prove that selenium and vitamin E weren't the answer. And at some level, easy for me to say in hindsight, but that really wasn't that much of a surprise because the data leading into it wasn't all that strong to begin with. But again, something that was done. So with that in mind, cancer prevention agent development, as summarized in the top here, our goal obviously would be like therapy development, like studying a new antihypertensive. You'd like it to originate from biological data or discovery data, or in the case of carcinogenesis, science that leads you to want to intervene against that aspect of carcinogenesis. So that's the goal. And then if we think of early phase studies, and that's the main thing I'm going to talk about because that's what our consortium mainly does, even though we've also done phase 3 studies, as you have as well. I'm really going to focus on phase 1 and phase 2. But the goal of those, obviously, are to find tolerable, safe agents that produce some sort of significant biological effect that foreshadows that they might, in fact, be of value as a cancer preventative. And as summarized below here, those are some of the issues we deal with. And I won't get into it, but even our phase 3 studies in cancer prevention, the struggle is what should be the primary endpoint. It would seem it should be diminishing the incidence of cancer. Well, our prostate studies even raise that question. Is that really the right primary endpoint? So all of that, especially in the development standpoint, really centers on this kind of phenomenon. And if we first start with how cancer prevention agent development is the same or different than cancer therapy, it really comes down to what I'm going to call surrogate markers. When you get right down to it in cancer therapy, and that when my career started, that's really what I did for the first decade or so, was cancer therapy development and drug resistance. It really is based on the surrogate that when somebody's tumor or degree of disease shrinks or changed, that really is the surrogate for the marker the endpoint you're most interested in, which is living better, living longer. And to be blunt, it's a pretty good surrogate. It correlates. Now, if you think of vascular disease, hypertension, cardiovascular disease, the really holy grail there is the fact that you have a non-invasive, predictive, quantitative surrogate markers of disease, that being blood pressure readings or lipid levels. And as a cardiologist told me many years ago, and I'm sure the same here, vascular prevention did not really take off until it became the therapy of lipid disease or the therapy of high blood pressure. That even though we do those things predominantly as a preventative, how they got used in our society is its therapy of hyperlipidemia. So it would be nice in prevention of cancer if we had that. The quick response is we do not have that. But we have things that we try and we look at. And work that's done here by many of you are examples of that. But intraepithelial neoplasia, tissue biomarkers, and we'll give some examples of that. And again, the real issues with the surrogates, or to be more precise, the potential surrogates that we use are that, one, they're not proven as surrogate markers yet, and we have issues and, frankly, at times unestablished issues of variability and sensitivity related to the biomarkers that we use. So if we start with just intraepithelial neoplasia, why or how would that be our kind of biomarker of choice? Well, as listed on this slide, it gives you kind of some of the reasons and examples of why it might. Certainly phenotypic and genotypic changes with later invasive disease, the more we study it, the more we find that they're not that dissimilar. That certainly the more we've done genomic analysis, whether it's of normal to abnormal to invasive disease, that those genomic studies really tend to show that there are some sentinel genetic events that carry forward, and as more and more of those genetic events accumulate, you eventually lead to the invasive disease, whether that's P53 as an initial event or other aspects of it. And as I showed on those earlier slides, intraepithelial neoplasia is already considered a disease by many. Certainly in dermatology it is, Barrett's esophagus, etc. So there are many treatments, and there's millions of dollars or more spent treating intraepithelial neoplasia. But again, the issues with our kind of system are what follows there. Variability in diagnosis and interpretation. It's important. There can be quite a bit of difference of what one pathologist calls dysplasia and another one does not. A key thing is slow and relatively low rates of progression to cancer. The more we study other tissues, the more we realize that there is dysplasia that leads to invasive disease, that there's field effects that you see. What we also know is the more we study other tissue types, the more we know that a lot of that dysplasia will spontaneously regress. We've known that with actinic keratosis. We've known that with cervical dysplasia. Adenomatous polyps. Work done at Wisconsin, work done at other places where they follow it, whether it's through CT colonography, others. A large number of what we presume and sometimes are diagnosed as adenomatous polyps, especially when they're small, will remit on their own. They don't all just absolutely progress. So there's a lot of issues with it, but it is, pardon the term, sometimes the only game in town when it comes to cancer prevention agent development. And here's just examples of intraepithelial neoplasia that many of the researchers here and myself and others have used many of these as kind of our targets or our primary endpoints, regression or changes to this intraepithelial neoplasia. So again, the preceding slide details, again, both the pros and the cons of that. Other things that we're looking at when we think of cancer prevention agent development studies are these biomarkers. Again, stressing none of these have been proven to be the holy grail of our surrogate markers, but nevertheless, again, it's based on growing knowledge of the biology of cancer progression, growing knowledge of just how risk and stratification of cancer risk are. But germline mutations and risk, I'll mention a few things at the end about that. Certainly tissue signatures, more and more, whether it's Frank's work, Steve's, others, are looking at what is going on genomically in our target tissue, both at baseline and in response to an intervention. Epigenetics, and I'll show you some examples of that. We use a lot of non-specific biomarkers, proliferation. Again, at least in animal models and others, if you are anti-proliferative, you usually are anti-growth, and that usually means that you can prevent or lower the incidence in animal models of development of tumors. And there's any number of ones that we look at, Ki67 probably being the most common. Apoptosis, again, the thought and goal and evidence that we want more apoptosis in the tissue, not less. So we look and wonder if there's pro-apoptotic effects. And that some of our studies, some that I'll mention, some that I won't, look at very specific biomarkers, targeted approaches, whether it's EGFR, phosphorylation, et cetera, insulin-like growth factors, AKT is another downstream. Certainly prostaglandins and cyclooxygenase have been a target for years. And again, those then can be the biomarker you look at. So in the context of that, and to give you a little bit of a description of this chemo prevention consortium that many of you have participated in. So based on this prior kind of discussion, the National Cancer Institute, Division of Cancer Prevention, decided in the early 2000s that they wanted to try and centralize this process a little bit more in cancer prevention agent development. So whether they would admit this or not, they basically were emulating what had gone on in the therapy group, or CTEP, starting more than 20 years prior to this, actually about 35, 40 years, with Phase 1 or the Decision Network, which was back before industry was really thickly involved in cancer therapy. A lot of it originated at the NCI. So one of the things, as many of you know, there's not a lot of industry involvement in cancer prevention agent development, so we're really kind of on our own. So the NCI organized this and wanted to centralize it, as stated, and eventually competed and selected 6 consortia led by these institutions, and we were fortunate enough to be chosen of one of those 6. And our initial consortia members are listed here, Minnesota being front and center, and the others as listed. And we did these studies to begin with. And even as we show kind of the studies and the maturation of the types of studies, you'll see that what we started with, and we still do, are nutrient-based products, things like that. And as we go on, you'll see kind of an evolution into vaccines, into repurposed agents, et cetera. But these are just some of the examples of the studies we did. Again, this consortium is based on doing Phase 1 and Phase 2, not Phase 3 studies. That's left up to either cooperative groups or other U series awards, et cetera. So some of the studies done, including Frank's Pioglitazone study, is listed here, and I'll describe a bit later. So in 2012, the NCI thought, and the Board of Scientific Advisors thought, that there was enough progress with this model that they recompeted it. And in 2012, we were fortunately, I should say 2012, one of 5 selected. And here I want to give kind of an anecdote. As many of you know, when it comes to dealing with the NIH or the NCI specifically, when they say the term negotiate, be careful. So one of the prior 6 decided they really wanted to negotiate the budget of this contract. Well, they negotiated themselves right out of the contract because the NCI said, here's the negotiation, which is the NCI term for take it or leave it. And they thought they could negotiate, and they were wrong. So the rest of us weenies kind of went with what they gave us. So anyway, so that's 2012. In our consortium in 2012, we made some changes, again, Minnesota being front and center, and added a few others to our group. And it's led to these studies, some that are complete, some that are ongoing, and the ones in red are studies we're waiting to do. Topical green tea is a study based on Paul Lambert's work and Hassan Mukhtar's that we're actually taking to Asia to do it there as kind of developing a site. Part of doing that, besides having more of a global impact, is these studies, as Frank has alluded to, and as you all know here because you've done many of them, they're hard to do. And so the greater your population that you can work on, the more likely you are to be successful. And there are some centers we've engaged in Asia that even cancer or dysplastic areas that you would consider to be relatively low incidence in Asia, they're so centralized and their numbers are so large that they dwarf almost every hospital in our group in terms of the numbers of eligible potential subjects. So we are looking to, for their benefit and our benefit, really do that. RG1-VLP is actually an interesting, new, novel HPV vaccine. As all of you know, and we've mentioned before, the current HPV vaccines, which again are very effective at the genotypes that they're designated are, is an L1 or late 1 protein derived virus-like particle. Well, this is based on a bit of L1, but mainly L2. And now L2 is less immunogenic than L1, but L2 is conserved. So in the early studies, this vaccine is immunoreactive to every genotype. So we are hopeful and interested in that, and that's work originated out of Hopkins and the University of Vienna that we're poised to do. So recently, again, thanks to Frank and many of you, we recompeted that, and we're waiting to hear. And it's gone from a contract mechanism to a U series. So I guess I don't have to be quite as careful with the negotiation. There won't be any facade of negotiation. It'll be, here's the budget, take it or leave it. But our consortium, if refunded, would include these institutions as listed here, and some that I've mentioned. And historically within the group, these are the types of studies we've done. And the ones in red are either new areas or areas that I hope that we, if refunded, will do more with. And then to describe it in a little bit more detail, here's how this consortium works. It did this way on the contract, and it would be this way going forth. In that, the advantage for you as an investigator is, to be honest, I probably should use better terms, but it's a shallow pool of competition. In that there's only 5 consortia, and we all submit, again, our ideas from within our groups. And oftentimes you're only competing against yourself, sometimes with solicitations where the NCI says, we want to do a colon adenomatous polyp study. Maybe there'll be numerous applying. Or we want to look at lung carcinogenesis or other things, or breast or what have you. So we'll compete against the other consortia, but as you see, there's only 4 other consortia. And oftentimes you can also submit, in the old days, what we would call unsolicited, but they have a different term now, but essentially your own ideas. And the NCI will either approve it or not. And it isn't because we are savants with our protocols. It's just to give you a sense of how relatively easy it is. About 60 to almost 70% of what we submitted gets approved. And each time we submit, it's more or less like putting in a 10-page grant. Budget's included, all the work is done, the biomarker, everything. There needs to be some modicum of preliminary data to support it. But so that's the initial process. And we essentially function as a coordinating center of a cooperative group. So a study that originates here at the University of Minnesota, that's led by the University of Minnesota, and I'll give you some examples of that, is indeed done here. But we're the coordinating center, and we work with the PI, whether it's Frank or someone else. And basically, whether it's some other cooperative group you've been involved with, we are the coordinating center for that, and then work and kind of buffer you from some of the issues related to the NCI. Now, the good and the bad of it for you as investigators are, as I was describing earlier, the positives would be, you know, the amount of preliminary data or the vetting that you need to do is certainly less than trying to get your own R21 or R01. The positive is the budgeting tends to be considerably better than an R21. Most of our clinical trials probably have a budget that ranges from a low of $500,000 to a high of over $1 million. The negative is that it's not as good as an R01, and I'm not going to try and pretend to any of you that it is. I mean, having your own R01 is still probably a better deal both for the institution and for you as an investigator. But if you're not sure you're going to get an R01, we are a mechanism where you can get preliminary data to then go after your own R01 or U-Series award. Now, the NCI is very involved. Unlike a grant, if you and I submit one of our grants and say we're going to do a translational project, preclinical or clinical, you know, once you've got the grant approval, it's kind of you do what you're going to do, what you said in the grant, whatever nuances you might make. With this, the NCI is much more involved. They have final approval over the protocol down to the periods that we put in it. And I kid you not, we argue about where periods should go in the protocol sometimes. We've stopped arguing about it, so we've followed their lead relative to that. But so, and that's a good and a bad thing. But those are some of the things that go in. The time frame I was describing earlier, if we put in a proposal and it gets accepted, usually it takes about a year maybe a little more than a year before the study's actually up and running. So it's not the quickest mechanism in the world, but it's certainly quicker than cooperative groups, et cetera. So that's the data is listed here. And if we just think of, again, chemoprevention development, this is just a summary to describe for some of you phase 1 and phase 2 prevention studies. When we look at prevention studies, not unlike a new antihypertensive or what have you, we might have normal volunteers, we might have people at risk. We sometimes can do dose escalations, sometimes not. And I'm going to give you some examples of studies we've done, and again, kind of stressing where the struggles are related to these studies and then what we do with it. So, with that in mind, I'll first start with this agent. And this is certainly a more, from my standpoint, exciting phase 1 study, in that this is a study of a novel rexinoid. And this originated from one of our partner institutions, in this case, the University of Alabama, Birmingham. And a biochemist there and synthesist by the name of Don Muccio basically was interested in developing novel rexinoids. And the key with this agent, which you can either call UAB30 or 9C UAB30, is it's an analog of 9-cis-retinoic acid. And the key thing is shown here is a tetralone ring has replaced the beta-inine ring. And again, as with all rexinoids, you got your ring structure and your prenoid groups off it. And this UAB30 the same way. And the reason they did that is they were very interested in trying to come up with a less toxic rexinoid. So rexinoids being hopefully reasonably preferentially binding to RXR rather than RAR. And then in this case, what they showed, and they had some nice work, and the references are listed there, that when they manipulated this structure, in this case a tetralone ring, it changed the heterodimerization. So just to remind you, whether it's PPAR gamma or thyroid receptor or vitamin D receptor, or in this case, retinoic X receptor, it has to do with dimerization before you get into then tertiary structures and before you get into DNA response element binding, and they then transcriptional control that it works. Well, what they nicely showed was that they could change and move the heterodimerization more towards homodimerization, RXR, rather than RXR liver X receptor. And they showed some nice work with that. And again, one can imagine that, again, the tertiary structure is the reason for that. So we then did an early phase 1 kind of pilot study, just looking at dosing and how we wanted to do the actual phase 1 study. And that led to this phase 1 study that, due to the vagaries of the NCI, and literally this study got caught in the last government shutdown because, again, we're a contract. We've had different times where we've succumbed to some of the political winds of the federal government. So this phase 1 study, which should have been one protocol, ended up being anywhere from 2 or 3 different protocols over numerous years. But the take-home parts are, we designed this in such a way where we would give a single dose, then a 7-day washout, then 28 days continuously, starting day 8, and then a final 7-day washout, because there are numerous retinoids and rexinoids that can alter their own metabolism. So we wanted to assess that in addition to looking at tolerance. And just to remind you, so 9-cis-retinoic acid, if you can get in 20 milligrams a day, you're doing pretty well because of toxicity. Tagretin, which I showed up there, which, again, different RXR, again, you can probably give anywhere from 300 to 600 milligrams a day in therapy with a lot of toxicity. So with this, we have actually gone all the way up to 240 milligrams a day with hardly any toxicity, and I'll show you a little bit more with that. But this is just a lot of data looking at the pharmacokinetics. And because of the prolonged half-life, we did see the AUC gradually increase from day 1 to day 36. But we did not see evidence of its metabolism being altered. And we saw linearity from 0 to 160, but we did not see it from 160 to 240. That either could be because we're overwhelming the system. It could be there's some data. These were 20 milligram capsules, so we were giving them a boatload of gelatin by 240 milligrams. So there's some evidence that gelatin can inhibit absorption. So we're trying to do some other things with that. But as summarized there in the data, But when we looked at the adverse events, grading, one of the things you'll notice if you follow kind of from the upper left to the lower right, you'll see a slightly greater incidence or evidence that maybe there were more grade 2 and 3 events as you got into higher doses. And what that was hypertensive events. Interestingly enough, skin toxicity, headache, nausea, other things like that that you associate with vitamin A derivatives, we didn't see even a hint of. But we saw hypertension events. But that was probably more related to how we did the study, the nature of how long the studies took, because I put this up here, one, to make your eyes bleed just from all the data there, but it's all the blood pressure readings. And the keys are here that there's baseline. This is compared to baseline, so systolic and diastolic look the same way. And placebo's the dark black here. And again, it's really kind of day one, multiple measures, then weekly, and then treatment stopped here. But what you really find there, and there's p-values, that there really was no evidence of any increasing blood pressure. It was really more a function of how and when we were doing some blood pressure readings. Some of those events actually occurred on day one before the drug was given. It's just the fact that blood pressure readings can be all over the place. So the actual hard data didn't show that. And then just to give you an example, the triglyceride levels, again, same thing. Placebo's in black here. The really keys are here to here. This is off treatment. And again, no evidence of even a gradual increase in serum triglycerides. People's triglycerides, when you measure them weekly, they're all over the place. But again, no specific trend with that. So when we think of this novel rexinoid as listed here, again, a lot of our studies, we do blinded, placebo-controlled, and the data I just showed you is a good example of why that's of value, trying to sort that out toxicity-wise, but also some of the biomarker aspects of it. So we saw little to no toxicity. That is both a good and a bad thing. As somebody who's done drug development for decades, over the years, I've given some drugs that had no side effects because they weren't doing anything. They weren't even binding. We've tried to do some studies on skin and other things to try and look at that. We weren't really able to show or not. So I'm going to hope like heck that UAB30, and again, there's evidence to support it, is in fact just that much better of a compound than 9-cis-retinoic acid that you can give 160 or 240 milligrams a day, because it has the same potency, the same ligand binding properties as 9-cis-retinoic acid at a molar equivalent. So it's not less potent, it's equal, if not even slightly better. So we are hopeful. And there's an ongoing breast prevention study, some skin and other things going on where we're going to look at more, obviously, downstream, oftentimes proliferative effects to try and show that biological tissue effect with that. So many of you know, probably appropriately so, because of the way immunotherapy and immunology has continued to grow and expand as we follow it, and certainly in therapy of cancers, checkpoint inhibitors have been a really significant improvement. There's just no getting around that. So not surprisingly, immunoprevention, and not just with what we hope to be the success of HPV vaccines or limiting oncogenic infections, but looking at them from a tissue standpoint. So one of our collaborators is Nora Desis from the University of Washington, and she's been developing DNA-based vaccines, as have others, both here and at our institution. But so we're doing an ongoing phase 1 study of a vaccine that's DNA epitope derived. In this case is listed, and you can see the vector and others, that the 3 components being HER2, IGF binding protein 2, and IGF-1 receptor. And the reason for that is that when you look at DCIS, or both in people and models of the progression of dysplasia across breast epithelium, those are gene signals that are frequently overexpressed and seen as you develop, again, further genetic changes. So Nora had already done some work in some models showing effectiveness in an animal model as listed here. There's some ongoing work preclinically of how these vaccines potentiate rexinoid or retinoid effects because of some of the other aspects of what happens with RXR, both homodimerization and heterodimerization. So it's an area that we in the NCI and many others are very interested across many different tissue types. In this case, this is a breast study. But the study that we're actually doing is this. So we're giving it, and this is through the FDA, and you'll look at the population's different. The first phase 1 study was normal volunteers. This is a study in women who've had breast cancer. Again, that wasn't our choice. I'm not complaining. That was the FDA's mandate of where we start with this first-in-human study. And so, again, a lot of it's arbitrary. We pick out doses. This is not a placebo-controlled study. This is basically an open-label study. And we, Dr. Desis, others, we kind of guesstimate on what we think the amounts of DNA-based vaccine we want to give. And we're giving them in groups of 10. And in this case, and I have it in red, the endpoints are essentially safety and immunogenicity. And again, even when we get into phase 2 studies, the issues are what should be our endpoints that could foreshadow that we're going to have the benefit and the effect that we want. But in this case, it's immunogenicity. It's looking for type 1 more so than type 2 immune effector cell response. But that's some of the things that we look at related to the phase 1 studies. Phase 2 studies, again, tissue specific. Many of them have been done here. This just gives an example of some of them. And the ones in dark are the ones I'm going to give you some further examples into. So this is an example of kind of an initial study. This was a randomized, placebo-controlled study of genistein. And even though I have listed here genistein and estradiol because most people thought of isoflavones and genistein specifically as a phytoestrogen, meaning estradiol-like. But we were interested in it because of its propensity to inhibit EGFR phosphorylation. There had been both preclinical and some animal studies supporting this. So it was one of our earlier studies. And of note, again, is our design over here. And one of the keys, as many of you already know, with these prevention studies, it's access to tissue. It's how do we build a clinical trial where we can get access to the target tissue. Now, for the skin or other areas, that's less of an issue. Of course, if I'm the one getting biopsied repetitively, I'm not sure I would call it less of an issue. But for us as the investigators, a little less of an issue. But for other areas, it's a struggle. And so a lot of these studies, we build around planned standard of care events. In this case, for bladder prevention studies, we build them around treatment or discovery of either superficial bladder cancer or planned cystectomies, and that was the case here. So men and women who are about to undergo TURBTs or partial cystectomies were randomized to genistein, multiple doses, or placebo. And the interesting things here are, and again to give some examples of it, is one, not just the fact that these studies are sometimes difficult to accrue to, but this was actually a study where we had relatively positive results. And maybe a bad sign is the NCI didn't even know what to do with positive results because it was such an infrequent finding sometimes in our studies. But so what I want to show is in the describe on the right here, upper right. So this is phosphorylation. So the brown is EGFR phosphorylation. And in the uroepithelium, so A is benign tissue placebo. B is tumor tissue placebo. C is active agent benign, normal urothelium. D is tumor urothelium. And what we showed in good or bad ways, but more in positive, is that our lowest dose of genistein, more so than the highest dose, there was a little bit with the higher, but mainly a lower dose, of genistein had a significant reduction in phosphorylation. Now, one of the things when you think of primary prevention that you should raise or question is, well, that's nice that you're treating the tumor, but isn't the goal to prevent it from becoming a tumor? That one of the issues that we struggle with is how do we assess or where should we assess? And I'll show you some data later where more and more it's kind of looking at the field effect or peritumoral or adjacent normal appearing, which frequently is genotypically not normal. But that's some of the issues when you think of the studies that we do. The other part of this is we looked at a lot of biomarkers in addition, and none of those really had a signal at all. So was this kind of a by chance, vagary of finding, or was there something significant there with some of the things we're still trying to sort out? And we are interested in maybe combining it with, in some other ways, with other EGFR agents, and we're studying that as we speak. So this is a study of Dr. Andres, Pioglitazone, the development of these agents years ago to try and combat insulin resistance, and then eventually with the discovery of PPAR gamma, that pioglitazone is an agonist for PPAR gamma. And that steroid receptor family also showing evidence of that it has anti-proliferative effects. So work that Frank had done earlier in his own study showing some evidence for effectiveness led to us performing, with Frank as the PI, a multi-site study randomizing men and women with oral lesions, as shown on the right, with multiple visits, biopsies, etc., to pioglitazone or placebo for 24 weeks, with the primary endpoint being regression of the lesion. And there were multiple other things that were looked at as well. And the issue here was the study was initially going to be much larger. One of the issues we get into, and I'll describe more later, is just how we do our studies, what size, what should be the primary endpoints, even how we do the statistical design might need to be reconsidered because of variability. But the upper panel on the right is the placebo patients. So lesion slight growth is here. Lesion regression is here. And here is the active. And essentially there was an insignificant difference in response rate between study drug, 46% versus 32%. There wasn't a real strong histologic regression rate. It was around 10% or less. But other biomarkers had trends but did not meet significance. But it certainly followed the pattern that you would have hoped to see, whether it's proliferation, apoptotic indices, etc. So we certainly showed that it was safe and tolerable, that there was at least a little bit of a signal. Now again, obviously you could say that's a pretty significant placebo effect, but again it gets into the fact that things have regression, even spontaneously. But there was a hint of it, which has led to an ongoing study of Frank's, which is a combination of metformin and pioglitazone because of, again, the data and evidence to support kind of metabolic dysregulation and our goals and efforts to try and improve upon that. And then another study originating here that's just getting started as well is a study of Britt Erickson's, which this was looking at, again, exemestane and endometrial cancer. The participant population is as noted, and this is a study that's just ongoing. And the point I want to make here, and gets into the biomarker part, and again, work here at Minnesota with Dr. Skubitz and others, is trying to come up with better ways, less invasive ways, to look at the target tissue. In this case, again, we're building it around planned hysterectomies for complex atypical hyperplasia or low-grade disease. Again, it's a standard event. We can get access to tissue. So we ask women to take exemestane prior to their surgery. And not surprisingly, that is not an easy sell across, whether it's prostate cancer, breast cancer, endometrial cancer, whatever. But we work hard at it, and the people here and elsewhere work hard at it, and we get people on it, but it's a struggle. But if we could come up with better ways that could do it. So a credit to the work here at Minnesota is looking at tissue genomics through other ways. Even just looking at it, what a study being done in this trial is tampon recovery. So just collecting endometrial epithelium shed via the vagina and collecting it and examining it. Because again, however you get access to that epithelium, the issue is getting access to the epithelium. So if we can come up with non-invasive ways to do that, that would be especially valuable. So just some future directions that we're thinking about in the consortium as listed here. And I'll give some quick examples of each. There's more vaccines that we're looking at with Dr. Desis and others. StemVac is more of a vaccine that Dr. Desis is developing, more directed at hormone receptor or triple negative developed breast cancer, looking at more of the epithelial mesenchymal transition as the target, if you will. and combining with rexinoids. Our own Doug McNeil's got DNA-based vaccines in prostate cancer that are in therapy studies that we're interested in prevention, and the L2-based HPV vaccine I'd mentioned prior. We're doing a lot more in gynecologic cancer, not just the work that Britt Erickson is doing, but there's more and more work showing that the precursor for high-grade ovarian cancer is actually fallopian tube dysplasia, or called stick lesions, as mentioned here. So our own Dr. Patenkar and Lisa Berlet have some agents they're developing, and we're looking at potential ideas. University of Oklahoma has multiple ideas that they've used both in animal models, and we're poised to do some clinical trials looking at whether it's a heat shock protein targeted agent or even aspirin itself, given some of the data supporting its role in types of studies that we're looking at. Here at Minnesota, whether it's Dr. Wattenberg preceding Dr. Andre, looking at aerosolized approaches. Again, I don't mention here, but obviously your own work in tobacco carcinogenesis are multiple areas of interest. From a toxicity standpoint, trying to localize delivery obviously is one way to get around systemic toxicities, whether it's aerosolized or topical. There's cervical topical approaches and multiple listed here. Dr. Betty Steinberg, who's one of the leading HPV researchers, mainly in pulmonary papillomatosis, is also developing for cervical cancer a topical approach that has some very strong preclinical data. In this case, it's all natural, for what that's worth. Curcumin, green tea, resveratrol in a topical approach with the data shown here. So that's something that its IND is essentially being approved here shortly in a study we're interested in doing. But one of our struggles, again, is tissue biomarker variability. And some of the things we're trying to do to look into that, in this case in prostate cancer, our pathologist Wei Wang is looking at us cataloging across about 40 to 50 prostate cancer specimens and prostate specimens. Just what is the variability of the markers we look at? So to be honest, in most of the tissues that we work in, nobody's ever cataloged whether regional differences, intra or interpatient variability. So when we do our designs, we are a little bit guessing. And sometimes we guess poorly, to be honest. But if you look here, so prostate samples, tumors 4, near adjacent 1, slightly further adjacent 2, distant is 3. And if you look just at proliferation, excuse me, across some of our early preliminary work, and there's quite a few samples to go, not surprisingly to many of you, you see that adjacency matters. Even if phenotypically it looks normal, it may be that genotypically it isn't. But proliferation varies and does correlate, we think, from this is epithelium and stroma, from tumor, close to tumor, slightly far away, distant. And we're doing more with that, but also just to catalog better just the variability of these, whether it's androgen receptor expression, Ki67, other things, because it may change how we do our designs. I am very concerned that we are setting ourselves up repetitively to fail because we need to do a 300-person study to detect a modest difference. And it may be that a modest difference is effective, because the way our studies are right now, we're only going to find very profound effects because of the variability involved in the fact that most of our studies are under 100 people. Other future directions include here, this is work out of Hopkins that's interesting in breast epithelium, looking at quantifying methylation or epigenetic effects. And this is good data, as listed here in the references. Again, as a potential tool. Again, as I said in the beginning, our struggle is what are the endpoints that we should be looking at that really point us in the right direction? Or is it germline issues with stratifying populations? Whether it's metabolism, obesity, here's work out of Penn State that's recently joined us, or germline issues with metabolism. There's a lot of people who felt for years that some people get benefit from the environment or natural products or even how you get exposed. Just like we know there's germline issues with tobacco carcinogenesis and metabolizing, there can be metabolizing issues germline in the positive or the negative. And this is also some work out of Hopkins where they wondered about that related to SOD2. So just in closing, and not just because there's many of you who I know are very interested and committed to this area already, but you know, eventually industry, others will take notice and get involved, but it's probably not going to happen until we figure out, again, surrogate markers, biomarkers, or better risk stratification, because the better we can identify our populations, obviously, the better our event rates are, the more we can learn, the quicker, the more efficient we become. And because of those things, again, eventually, whether you consider it a good or a bad thing, but the more people interested in prevention, the better. In this case, industry. It would be nice someday if they were more interested. But prior to this, since we were only good at killing their drugs, they've tended to stay away from us, whether you remember the rofecoxib story and celecoxib story. And I've got fear in red there because we all know that our public, our community, you know, when we grew up or when I grew up, the fear was I'm going to drop over from a heart attack. And people didn't make the correlations to risk. It's just some 40, 50, 60, 70-year-old just drops over. Now what's kind of replaced that, in my view, is that people have a fatalistic view of cancer, even though we all know there are many factors that contribute to cancer, environmentally and otherwise, that you can avoid, or behavioral things. But most of the public tends to have a fatalistic view of cancer. And I have no doubt that when we come up with proven effective agents that lower the risk, and as we better identify who those populations are, that public fear, and I'm not saying that's a good thing, will lead to industry really pushing this much further, so that many of us, you know, the work that we've been working on will someday kind of see the light of day, if you will, with that. So this is just the acknowledgments mainly of the University of Wisconsin team. Also thank the University of Minnesota people who have been involved in the clinical trials, and there's others besides Frank, Bev, and Britt. But with that, I'll close, and I thank you for your time and patience. Thank you.